Connie Brewer. I'm a professor here, just down the hall on this floor. And it's my pleasure to get to introduce our first speaker for the summer series, Herr. Alan has been at UW for about 11 years. But his connection to the Northwest goes way back. He's from Boise, and he tells me just now he was a cowboy in Boise. He received his BS in biology at PLU, and after graduating, he went to the University of Utah, where he got his PhD in human genetics. And he worked on a really interesting basic problem in molecular and cellular biology, where he was interested in how ribosomes are able to jump around on a message, which was really unusual. He then finished that degree and went to the UK, where he switched organisms, where he now worked with one of our common pests in the plant world that is a very famous plant called Arabidopsis. And he worked on an interesting problem in this organism, which was the ability of a genome to silence the transposable elements using an RNA intermediate. And so he worked on that for a while. And then he moved back to the Northwest, to the University of Washington, where he was a senior fellow in the pathology department. And again, he shifted gears to start working on a really interesting problem. He's picked really interesting problems in the basic area of molecular biology and genetics. And this one was to try to understand how come some cells accumulate mutations faster than other cells, and some cells are really good at resisting the process of mutation. And that has to do with having mutator genes and anti-mutator genes in our genomes. So some of us have the propensity to accumulate mutations during our lifetime a lot faster than other people, and that can lead to things like lifespan problems or diseases and just overall fitness. So he has been working now in his own lab. He's now a research associate professor in pathology, and he has his own lab that he runs, and I've been able to work on several committees with Alan. That's really how I met him. As you can see, he's worked on a wide range of organisms, from bacteriophage, these viruses that infect bacteria, to plants, to human cells, and then to the pinnacle, to yeast cells, which is what I work on. But I don't think he's going to talk about any of that tonight, all of that work that he's done in the lab. Instead, he's going to tell us a very personal story about lung cancer. When I was asked to organize and invite speakers for this summer series, Alan was the first person I thought of. And so I'm really delighted that he accepted my invitation to come and give this very personal story. His story is really one of great courage and resilience and optimism. And it really deals with the intersection of biology and genomics and cancer biology. A little bit of self-experimentation, perhaps. And an awful lot of collegial collaborations with the people in the community here. So I'm really excited to introduce Alan. Be prepared for a really inspiring and awesome talk.
No pressure. Well, no pressure. Thank you for that incredibly kind introduction. Let me drink some water. There's always the possibility that I'll drown. Okay, well, I guess that's not going to happen, so I guess I have to talk to you. So, I have cancer. That's a sentence you probably heard before. I'm not the first person to tell you that. In fact, there may be people in this room who've said that same thing. There may be people in this room who have had loved ones who said it, and you still remember that. You may say this someday yourself. Sorry to be a downer, but it's the world we live in. So how big is the problem? This is a lifetime risk of cancer. So the line to pay attention to if you're just sort of worried about your ability to escape is that actually odds are you're probably going to have some kind of cancer before you die. I don't want to leave you there, of course. I think that there's really no better time in history to have cancer, so you might as well. there's so much that's going on that's to be excited about. And dividends of countless years of research is paying off in some interesting ways. What I would like to begin with is not about lung cancer, but I'd like to begin with tennis balls. So what does N mean? Because there might be some people who are not thinking about numbers on a regular basis. And I don't want to assume anything. In fact, I'm going to assume that you're infinitely ignorant, but infinitely intelligent. So I'm going to keep dropping these balls. And the more I do this, you start seeing I'm getting a distribution over here on the right. So one more time. And I'm going to pose a question to you. Which tennis ball would you take to Wimbledon? It's a trick question. Actually, it's a pretty easy question. So you're going to say, I'm going to take this pen ball on the left over here. And that would be a perfectly good answer, except for the fact that this is just one ball. This is not a whole brand of balls. Really, the question we should be asking is, what brand of tennis balls should we take to Wimbledon? Because this was an N of 1 with 5 replicates. We just bounced this ball again and again and again. And it tells us a lot about that ball, but it doesn't really tell us about the brands. How good are they? Would you want to trust your center court aspirations to that? So a better experiment would be to get 5 different cans, freshly opened, and you'd just keep dropping them, and you'd measure. And maybe, just possibly, with those many replicates, you'd start to see a difference in the mean height that those balls would bounce. And you'd think, okay, I can determine whether they're statistically different. So what are some of the key assumptions with this kind of study? We're going to assume that these cans were fresh from the factory, they didn't sit around, that they didn't have any leaks of air in them, so that they were really the best efforts for each company. And from there, we can make something about some sort of statistical conclusion. So what about patients? I don't know if you guys can see this down here in the back, but that's red. It says, what about patients? That means it's important if it's in red. So patients. Well, we're not all alike, are we? Just look around this room and you'll see some people have long hair, some people have dark skin, light skin, some people are male, some people are female, some people have green hair. Hello, Ian. So the way that we treat cancer or go about finding treatments is that we take this population, and we know that they're different, but we try to get them as sort of uniform as we can, and then we separate them into 2 groups with N's that were sufficiently large that we'd have enough what we call power to tell statistical differences. And then we do a treatment A versus a treatment B. So treatment A would be standard of care, something that you know works. Treatment B would be some sort of experimental treatment. And lest you think that we're going to give you something horrible, usually experimental treatment can mean standard of care plus something else, or it's going to be something that, we've got good clinical evidence to think that this is going to work. Does that make sense? So then what we do is in cancer, we look at something called the fraction with progression-free survival. This is one way of representing the data. And so the purple line is our experimental treatment, and this darker blue line is the standard of care. And so given the means between those two, the medians, we might think, yes, we've got something. And that works for the large population. But hidden within this population are patients that are, they're different. I mean, it kind of reminds me of Life of Brian, when the guy says, I'm different, we're all different. I'm not. Yeah, well, anyway, I got that one totally botched. But anyway, so here's a person who is different. And let's imagine this represents a group of patients that are different. And they might get shunted both into treatment A and treatment B, just like we planned, but the outcomes of those patients are different. So this gold patient is one of these patients that responds really well to this treatment. And they live for a long time, durable response. So really, what we love to do in the modern approach to cancer biology is to be able to sift through these patients and bin them into different groups, not by the color of skin, of course, but by some genetic trait of their tumor, perhaps, or something. And then we would be able to tailor these treatments and get long, durable responses for everybody. Yay! So what do we have to know in order to do this? And again, I'm going to assume you're infinitely ignorant, so bear with me. This is called DNA. It is a molecule that's double-stranded. It has two strands of DNA which are elegantly wrapped around each other. And they're connected by these base pairs that join them together. There's T pairs with A always, and C pairs with G. And this structure is the foundation for life. What's remarkable about this is that all life, except for maybe some RNA viruses running around, utilizes this molecule to exist. It's one of the most profound evidences for evolution. So we can represent this as a string of letters, which makes it a whole lot easier to see. And just to get you a sense for how big the human genome is, I want you to look at this massive slide. So on the left over here is we have a representation of each of the 22 chromosomes and 2 sex chromosomes. Now there's just one of each of these autosomes, as they're called, 1 through 22, and then there's the X and the Y. So I know you're thinking, Alan, they're not that big. It's only 259. Well, that turns out million base pairs. So the size of the human genome is 6 billion base pairs if you put both genomes, one for mom and one for dad, together. So it's enormous. Each one of these chromosomes represents one continuous strand of DNA. It's quite remarkable. There's something like 20,000 protein coding genes within the human genome, and they're sort of embedded. You wouldn't be able to see them unless you knew the code. And so what we know about the code is that genes start at ATG codons almost universally. There's a few exceptions. And they stop at stop codons, which I'll show you in a second. In order for a gene to be expressed, it has to be transcribed, and that little wiggly green line across the thing is the action of transcription, which magically gives you a messenger RNA. Now, RNA is very much like DNA in terms of structure. That's why we call it transcription. It's a copying process, except that T has been replaced with U, and these are not deoxyribonucleotides, these are ribonucleotides. So within this messenger RNA, and I'm missing a few things for the aficionados, but we can recognize methionine codon and we can recognize the stop codon. So how do we know this? Well, this was worked out many, many years ago, very carefully in the field that I did my PhD thesis in, the field of translation. And so every single triplet nucleotide within the code has been assigned a meaning. And so, there are some codons that all code for the same amino acid, in this case, proline, or maybe threonine. And then there are some which are sort of off by themselves, only tryptophan. But this is quite amazing. I know it could be that this seems like old hat high school biology, but for a moment, just pause and wonder at it, because it is amazing. So this also turns out to be important for understanding mutagenesis. So before we get there, in order for that information to be realized in terms of protein, it has to be converted to a different kind of information. We call that translation. It's the process of protein synthesis. And I don't know if you can see it there in the back, but what we have is a beautiful couple of eggs representing a ribosome. And these squiggly blue lines represent transfer RNAs, which are RNA structures that fold into a double-stranded molecule that presents an anticodon which decodes each one of these codons. When they do this in the context of the ribosome, they sequentially bring in amino acids that are covalently attached to the 3' end of the tRNA. And then something magic happens, which certainly goes back to the dawn of time. It's called peptidal transfer. And this growing peptide chain is transferred to the new incoming amino acid that's on this tRNA here. And then the ribosome moves. And it does it again, and it does it again. For proteins that are enormous in size, thousands of amino acids long. Again, remarkable. So this is what proteins might look like. This is one of my favorite proteins. It's DNA polymerase delta, and it looks like a bowl full of spaghetti. But again, this is a single molecule that is simply twisted and turned and wound together. There are structures like this, which are called alpha helices, and in the middle, beta sheets. If you were to look at this in its full form, it looks more like this, space-filling molecule. There's really not a lot of gaps. So this molecule, DNA polymerase delta, copies DNA. It's essential for life, for accurately copying the genome. And what I want to just point out is how neatly this thumb here grabs one of the strands of DNA. So these protein machines are capable of exquisite selectivity and contours that are important for their function. So what about genetic misinformation? Mutations. So when there's a change in the DNA, that occurs on both strands and for it to become fixed. And when that happens, the messenger RNA is also different. And that now codes for a different amino acid. So in this case, we've changed the nucleotide at the front end of a triplet, what's also called a codon. And that means that a different tRNA decodes it. And so now we have a protein which is a mutant protein. And we use a little jargon in biology where we say what the wild type or normal amino acid is, the position number, and then the mutant afterwards. So L5V would be what this would be. So sometimes you lose a base. This is an example of a frame shift. And what happens is now the ribosome reads along to a certain point, but it actually reads in a different reading frame. It's almost like if you had War and Peace and choose your favorite book, Huckleberry Finn, overlaid on top of each other, two novels, meaning totally different things, but the sentences were all there if you just knew how to read them right. So at any rate, when this happens, the normal protein sequence ends up with a tag that the cell never sees, has never seen before. This is not normally translated in cells. And that turns out to be quite important in the context of cancer because these end up being a source of differences between normal cells and cancer cells. Another type of mutation that occurs is something called structural variants, and these can be deletions, where double-stranded breaks flank a gene, and this gets joined by a process called non-homologous end joining that leaves a junction within the DNA, a scar. It can also be something called a translocation, where breaks occur on opposite chromosomes, and then what happens is they switch partners. And so now what you have is one chromosome attached to the other end of another. This is called a translocation. And what can happen as a result of it is that the expression of genes can change quite dramatically. So in the above case, in the normal case, gene 5 is being expressed by a promoter element, something that drives transcription over here. When the translocation happens, this now drives translocation of gene 6. And so now a gene which should be off is now on. And this is one way that cancer can happen. So now I want to talk about mutations in the body. This is your body. Some of your mutations are inherited. They come from mom and dad. Thanks, mom and dad. Every single cell in your body has that mutation. Other mutations occur not because you encountered a bag of Skittles, but because you simply exist and mutations are arising in your cells. Sometimes they arise during the process of development. So they happen very early. And so you might have patches that are clonal mutations. You'd never know it, but the mutation is there. And other times, this is just the process of your cells turning over and dividing and turning over and dividing. So just to kind of help you think about what the consequences of that might be, think about the fact that your mom and dad gave you 2 genes. Maybe they gave you 1 mutant gene. We would call that a heterozygous mutation because the other allele, it's a fancy genetic term we use, is from mom, and it's perfectly fine. So the question you would want to know, is this going to affect gene function? So most mutations are recessive, or what we call mildly semi-dominant. They probably don't have a huge effect. Those heterozygous mutations actually are probably part of why we are as diverse as we are. But some are going to be dominant. And then if you happen to lose the good allele, now a loss of heterozygosity phenotype where this allele, which is normally silent, is going to be expressed, is much more likely. So that kind of gives you a think about mutations and their potential impact. When we think about cancer, we can think of sort of three classes of mutations. So one are oncogenic driver mutations. These are mutations that stimulate cells to divide in the absence of any growth signals, and they're generally dominant, not necessarily always, but let's just say most of the time. Tumor suppressor mutations, they lose the ability to regulate the cell cycle, and often in response to DNA damage. And they're generally recessive, but not always. BRCA1, I believe Tom's in the audience would confirm that that is sometimes semi-dominant. So those two mutations are the ones we think about when we think about cancer. Larry Loeb, who's in the audience, would also have you think about passenger mutations. So these are mutations which accumulate during just development of an organism, and also just during development of the tumor and growth of the tumor. And they can influence the resistance or susceptibility to treatment. So with all of these mutations and all of this cell division, it's important to think about cancer as an evolutionary process. This figure here shows environmental or endogenous mutations affecting some cell and creating a mutation which allows it to start dividing. And that mutation would be a driver, like in a little red triangle. And as that clone expands, it gets more mutations. Possibly early during its development, it might get a mutation that increases genetic instability. And if we know anything about life, is that if you're trying to evolve, the more mutations, the better. But it could also happen later during cancer evolution. It depends on where things, just random chance happens. Each of these little lines represents barriers to unrestricted proliferation. And you can think about this as just the constraint of a physical space within the body. It could be an issue of getting past a cell cycle checkpoint, or not having enough blood supply, which would be an issue of nutrition, or maybe not having enough oxygen. Each of these things can be overcome by mutation. Our cells have a contract with each other. They have a genome, which is, let's just say, perfect. And the agreement is that all these cells are going to work together and make a body. But in order for a cancer to evolve, all it has to do is mutate that gene and this gene, and then it can cheat and start growing. So it's eminently available for them in terms of evolution. So the person who came up with this idea of an early mutator is Larry Loeb, and I'm indebted to him because many years ago, when I graduated from college in '92, it was Larry who gave me a job. I mean, literally, I just put a resume in his mailbox and I got called for an interview. How about that? When was the last time you faculty members did that, right? Unbelievable. And I have stayed in touch with Larry ever since, even though I only stayed a year in the lab and then went off to graduate school. So key concepts I've learned from Larry is that every cancer is a unique genetic entity that is actively evolving. And we need to treat cancers as a collection of diseases with similar but distinct evolutionary trajectories. So, with that as a prelude, I'm going to tell you my story and a few insights I've picked up along the way. Oatmeal. That's it. Now, oatmeal. So, in 2012, someplace in here, it was early in the morning. I had a very large bowl of oatmeal. I make like half a cup of oatmeal in a very large bowl so it doesn't overflow in the microwave. And I was reading the paper and just on autopilot, and a flake of oatmeal went down my bronchial tubes. And I went, oh, that's not good. I said, well, if it's going to be a problem, it'll show up later, right? So I kind of just forgot about it. Someplace in here, I started feeling kind of gurgly in my chest. Like I could do this neat trick. Scott can vouch for this. I could go, and I get this sort of sound. It's like, that's not good. It's just the oatmeal talking. So my first little bit of wisdom is don't misinterpret symptoms in light of past events. If you have a bruise that shows up out of the blue, if you're feeling tired and you don't really know why, don't just assume, don't just interpret it in terms of like, this is the only thing that's gone, I'm fine. Pay attention to it. I would say don't be neurotic about it, but if you have something which persists for weeks, you should do something about it. A second bit of wisdom is to see a primary care physician regularly. Develop a relationship with them. This is mine. I didn't have it when I started feeling rough in my throat. This is when I had my first appointment with him, and I said, Dr. Reagan, I have this thing going on in my lungs, and I'm worried about it being something bad. I didn't actually say cancer, I just said bad. He said, well, hey, listen to me, you sound good. Well, I'll give you a chest x-ray if you want it. I don't know if you can get in right away now, but you can come back. 1, 2, 3, 4, 5 weeks later, I went and got an x-ray. And I can tell you that that walk across the parking lot to go in and get my chest x-ray was very difficult. We sometimes avoid doing things because we're afraid. It's better to know the truth. And this is sort of a come to Jesus moment, but if there's anybody here who has something that's persisting or something that you're afraid of, it's better to know. So this was the radiography result. So consolidation following posterior segment of the right upper lobe indicating pneumonia. Follow-up radiograph 6 to 8 weeks is recommended for completion of antibiotic therapy to document complete resolution. And I thought to myself, this can't be right. I don't have pneumonia. I feel great. I mean, I don't have any fever. I don't feel achy. This just doesn't make sense. So I talked to two of the smartest men I know. This guy over on the right is my dad. He's a cardiovascular surgeon, retired. Very, very intuitive. I said, Dad, I just don't get it. I just, he says I have pneumonia. I just don't think that's right. You should see a pulmonologist. I saw Larry the next day talking to Scott. In walks Larry. I said, Larry, do I look like I've got pneumonia? I just got an x-ray and it says I have, and he said, you need to see a pulmonologist. Listen to the voice of experience. Seek it out. Get counsel from people that are wise. So this is from diagnosis to surgery. This guy right here is Dr. Josh Bendit. It turns out that he is the son of Earl Bendit, who was the chairman of our department. What a small world. And it turns out that I had asked my primary care physician to book me an appointment in with the pulmonology department. And weeks went by, and Larry was worried. And I said, could you make a phone call? He made a phone call like here. And I was in to see him there. So it pays to know people. Don't be afraid of using your connections. It's important. So what came back after imaging with a CT scan and then and a biopsy a few days later, which I will say is one of the worst experiences of my life. That was horrible. Hose going down your nose, all kinds of things to keep you from gagging. Not a good experience. I'd tell them anything. Anyway, biopsy. So what it came back was that it was adenocarcinoma, non-small cell lung cancer. Standard of care is surgery for that type of treatment. If stage 1 or 2, and it says systemic therapy if 3 or 4. So we were set for surgery with really one of the best surgeons in the field, Dr. Doug Wood, who's here at the UW-MC, scheduled for the 9th, thoracotomy, upper right lobe resection. This is the pathology scoring sheet. This is not my lungs, it's an image of somebody's lungs. And what you can't see here are that in between these two areas, which is called the mediastinal space, are lymph nodes that exist. And there are lymph nodes kind of all over the place, but these are some key sentinel lymph nodes that they look at to see if the cancer has spread out of the lungs. And so the surgery was initially just to open up my neck and to take those out, to look underneath the scope and decide, yeah, he's good to go. Let's take out the lung. That's what happened. So Karen, my wife, who's here, can attest that was a long hour and a half or so. But so this is the right upper lobe. I don't have this anymore. It's gone. The pathology came back as a 4 centimeter by 3.5 by 3.5 tumor with no lymph node involvement. And according to this score, that leaves us with a stage 1B cancer. So what now? For this type of tumor, the probability that you'll be fine without any treatment is greater than half. I'm 43. I have 3 pretty small children, and I'm thinking about long-term consequences of exposure to chemotherapy, DNA damaging reagents. My mom, who had cancer, which I'll touch on briefly, had also kind of foregone chemotherapy, and I thought, I'll be okay. So that's what I did. This is a year later. So they took the entire lobe. They had good margins. I mean, that's what you learn about when you're a kid. Oh, they got good margins, it must be good. But nevertheless, I have about a dozen tumors on both sides of my lungs that are growing. So on our little chart, suddenly I'm way down here at stage 4A. And I just want to say that not all stage 4s are the same. My disease is caught very, very early. It hasn't metastasized to any other part of my body. It seems to like the lungs. I don't really understand why and how it got to the lobes that it's in. That can be a discussion for later, perhaps. But the key thing is this is where I am. So on to testing and treatment. So this is my amazing doctor, Renato Martins, who has become a very dear friend. This is Colin Pritchard, who runs the UW Lab Medicine and is also a dear friend and actually sits on the committee of my grad student. That's how good a friend we've become. So the thing that for lung cancer is that to know is that it's worth doing some genetic testing because there are 4 groups of patients that have mutations that have pretty good outcomes. So these patients carry mutations in the epidermal growth factor receptor, that's what EGFR stands for. They carry ALK translocations, ROS translocations, BRAF mutations. And then there's this other thing, pemetrexed super responders, which kind of are over here. But these 5 groups of patients do really well. And it would be great to be in one of those groups. So genetic testing. There are these targeted therapies for each one of these mutations that will give a patient maybe a year or maybe more before they get a resistance mutation. And that can be precious time. I was negative for all of them. So Colin did the UW Oncoplex, where he looked at 200 proto-oncogenes. When we say proto-oncogenes, it means they're not oncogenes right now. We think, well, they could be oncogenes if they had a mutation in them. They're something involved in cell proliferation. And so they have a way of pulling all those out massively parallel and looking to see if there's mutations. And there was a mutation, a subclonal mutation, which means it wasn't in every single cell within the tumor. It was in only some of them. It encoded an S33F mutation in beta-catenin. And actually, this turns out to be a pretty important functional mutation. So I know that you guys aren't all cell biologists, and I'm not either. So here is what's called the Wnt signaling pathway. And this is all worked out in Drosophila. So this just underscores the value of working with model systems. So in cells that are in a non-dividing state with respect to Wnt signaling, there's this strange thing that happens. And that is that beta-catenin is made. It assembles with these characters, GSK3 phosphorylates it, and then beta-catenin gets degraded. And this happens over and over and over again, so that beta-catenin levels stay low within the cell. When Wnt, let me go back over here for a second. So when Wnt signaling, when Wnt is present, it binds to this receptor, and it sort of recruits these guys, and that allows beta-catenin to go into the nucleus where it's able to do transcriptional stuff, turn genes on, get cells dividing. So in abnormal signaling, the S33F is in that site which gets phosphorylated, where that phosphate gets put on. And that prevents phosphorylation from happening. And so it doesn't get degraded. Its levels rise dramatically, and then it comes in the nucleus and competes off this grouchy protein called Groucho. And then cells are able to divide. So Rod Schmidt in our department, who's one of our retired pathologists, but is just an amazing guy who still kicks around occasionally. He stained the tumor for beta-catenin. And what you can see is the subclonal nature within this cell. So what you're looking at here are cancer cells. They're kind of columnar. They're the cells that line your airways normally. And so that's what they're trying to do. They're trying to create little tiny alveoli, or those little sacs that you take in your air. But of course, they're so packed in that there's no space. It's sort of laughable and sad. The mutations that have, or the cells that have a mutation beta-catenin are stained in brown, and they're more disorganized and packed in together. And this fits with the role of Wnt signaling in development, where cells become more disorganized and stuff like that. So this was quite a nice validation that this was a subclonal mutation. Alas, there's no targeted therapies for the Wnt signaling pathways, so chemotherapy it is. So I told my colleague, Matt Kaberlein, who's just down the hall from me, that I was going to be doing chemo. And he said, well, have you thought about doing Walter's thing? Walter's thing? What's that? So this is Walter Longo. And you know, I guess you could say that if you really make it as a scientist, you'll be known by your first name, kind of like a Brazilian soccer player. He was a rock star, actually, at one point before he became a scientist. So, you know, maybe it's true. So this is the thing that, that got my attention. So before I do that, let me tell you a bit more about what Walter, how he got to this point. So he's an aging researcher interested in stress, how cells respond to stress, because sometimes cells that are stressed ironically live longer. And so he thought, well, would they be more resistant to chemotherapy? He was thinking of how could you make the whole experience of chemotherapy less onerous for patients? And so they started with yeast cells, not really a patient, onto mammalian cell culture experiments, and then ultimately into mice. And what you're going to see is 2 mice that have been exposed to 10 times the dose of chemotherapy that you would normally give somebody. One mouse has been fasted 48 hours before chemotherapy. The other one has been fed normally. Oh, that totally steals the punchline. That's so depressing. Okay. So this is no fasting before chemotherapy. Neuropathy. It's not feeling very well. I think that's safe to say. This little guy, he's been fasted before chemotherapy, and he seems pretty happy. So which little mouse do you want to be? If you're going to go through this, it certainly gives you pause. Now, you got to think critically about this. So, okay, so you're protecting cells against chemotherapy, but what if you protect the cancer cells? Isn't that a problem? Because that would kind of defeat the whole purpose of fasting. Plus, it wouldn't be any fun. Well, we don't want more videos. We actually want to go away from here. Oh dear, there. Okay. So Walter was also concerned about this problem, and they published this paper in 2012, where they gave mice cancer in a pretty artificial way, to be honest. They had cancer cell lines. They injected those into mice, and the poor little mice got tumors. And then they did one of 3 things, or maybe 4 things. They did nothing. That'd be the control. They fasted them, those would be the blue diamonds. They treated them with chemotherapy, that's green, and red, they did both. And boy, the whole error bars look pretty good. Seems to be a synergistic effect between fasting and chemotherapy. But again, thinking critically about this, these are not what you would call normal tumors. They have been cultured in the lab for years. They live in a high oxygen environment, so they've had to adapt to that. And then we stick them in some place in the flank of a mouse and they form a tumor, but it's not like they live within a normal tumor niche and have all the relationships that would normally be there. So it's good to be skeptical of this data. So I called Walter and I talked to him for 40 minutes about this whole thing. And he shared this data with me, which was published a couple of years later, where they took mice that were lymphoma prone. They would normally get lymphoma late in life. And about here, 16 months, they started fasting them every other week for 2 days, and did that throughout. So their cohort that had been exposed to this fasting mimicking diet, which has a little bit more vegetables, but it's very low calorie, they had less lymphoma. They had fewer lymphomas when they were sacked. So there was something there. That was enough to convince me to do it. And so, this is the problem about being a scientist and being a patient. If you're a scientist, you can just do the experiment again. You can get more N, and really convince yourself. If you're a patient, you've just got one go at this. So you have to decide. So I decided yes. This is my current fasting chemo protocol. It involves cooked greens, onions, and olive oil for breakfast on Monday morning. And then a couple 100-calorie soups, noon and night, until the infusion day, which was several days later. It needs to be at least 48 hours in order to make sure that you're fully into ketosis. And then after at least 24 hours, then you can start eating normally. I have found that I cannot give up coffee, so I have coffee here. I also exercise because I found that it helps that transition from getting into a non-fasting state to being in ketosis. Just a little bit of exercise burns through all the glycogen in your muscles and you feel great. So there you go. This is the imprint of fasting over 4 years. So what you're looking at are two graphs. One is glucose along the bottom, and the other is something called anion gap. And this is a measure of the amount of molecules just coursing through your bloodstream. It's something that they always kind of look at. It's one of the things that are in here are ketones. And so each one of these little triangles is where I fasted. And the initial doses of chemo, carbopemetrexid, were here. This guy right here, I wasn't really well enough to fast. And so I didn't. So you can see my anion gap drops way down. But you can kind of follow through, and there's a pretty good correlation between this measure and when I fasted. And there are some breaks, which I'll be talking about. In terms of glucose levels, glucose, it's amazing how good your body is. You think, okay, I'm going to fast. I'm going to take my glucose levels down to, well, you can't live without glucose. Your brain actually needs it. So your body maintains this homeostasis someplace. I mean, even if you don't have any nutrition, it's getting it from someplace, so that's just impressive. Fasting in my liver. There were a couple things. So one of the things about fasting is you end up sort of catabolizing your body. One of the things you do is you take down your liver. Some people say that you might lose 20 to 30% of your liver during a long fast. So you might think that you might have elevated liver enzymes in the process of doing that. And you can sort of monitor that. I had some elevated liver enzymes, and then bam, I had this huge spike. And it turns out this wasn't due to fasting or chemotherapy. It was due to a common over-the-counter antacid, Zantac, because as soon as I stopped taking this, boom, down came my liver enzymes. The other time I had this was when I thought I would go all natural, and I took some turkey tail mushroom extract, and I had another bout of elevated liver enzymes. And my doctor said, "Are you taking anything else?" I said, "Well, I'm taking turkey now." "Stop it." And I went back down. So, caution. Combining fasting and drugs or natural compounds can have unintended consequences for the liver. Seriously, that's an important takeaway. So this is where we started with chemo. This is where we were more than a year later. And so it's basically the same section. It's not exactly perfect, but some tumors have kind of disappeared from the scene, and there's this little one here that remains. And what was striking about it is that the CT reports were coming back with things like findings could represent multifocal atypical adenomous hyperplasia. Well, basically what that means is that they knew I had cancer, but looking at the radiography, it wasn't obvious that, wow, that's a big tumor. Things had responded well. So we thought that was pretty cool. So why the gaps in the treatment? Turns out that this is due to side effects. And this is really one of the most important things when you start thinking about chemotherapy. It's what limits most patients from staying cancer-free, is just being able to tolerate the treatments. So over the course of my treatments, I've had more than 50 doses of pemetrexed. Early on, I started to develop maybe 6-7 months into it, I started to develop some saggy eyes underneath my beautiful blues, and the edema kind of got worse until I had an acute episode of fluid around the heart and lungs. Woke up with a lot of pain, and when we realized what was going on, we stopped treatment. And so I went for like 6 months without any treatment. And my tumors were happy about that. They thought that was great. So I got some regrowth, and they came back stronger than ever. Yeah, way to go. So the question is, what now? So at this time, immunotherapy was just starting to become a possibility. There had already been the trials, which were very promising, and people were starting to think about combinations to make it even more of a sure thing. So let me tell you a little bit about immunotherapy if you don't know about it. So our cells are constantly chopping up their proteins and presenting fragments of them on the surface using the MHC complex. And so, our normal cells are chopping this up, presenting these peptides. We don't have any T cells which recognize our normal peptides. But cancer cells produce some mutant peptides, and those get on the surface. And it turns out that all the T cells that would recognize normal peptides left the building when we were in development. And what's left are cancer cells, are T cells that recognize something other, not self, but other. And so what should happen is that T cell recognizes this peptide MHC complex and then absolutely destroys the cancer cell. So this probably happens early during cancer evolution, at least with some tumors. But eventually what happens is that this provides a selection pressure for cells that start overexpressing something called PD-L1. And PD-L1, turns out, is a repressor of T cell function, and it interacts with the PD-1 protein on the surface of T cells. And so that turns it off. This is what we call a checkpoint. And so checkpoint therapy works by giving antibodies that bind either to the PD-1 protein or the anti-PD-1. And the thought is that then the T cell can then destroy the cancer cell. So these drugs are in the market, pembrolizumab and nivolumab and avelumab. They have fancy names like Keytruda and all that stuff. But anyway, those are what the insiders call them. Actually, Pembro, that's what they call them. Anyway, so this was some really beautiful data that showed that pembrolizumab worked really well for patients that were sort of pre-screened for having high levels of PD-L1 in their tumor. And what you can see is that initially you don't separate from the standard of care, which is docetaxel, but then these patients have long durable responses, a subset, maybe 30% or so. So that was really encouraging. Why doesn't it work for everybody? That's an important question to ask. So there's sort of two hypotheses that you can come up with, and I'm sure there's more. I'm not an immunologist, but one hypothesis is that some cancer cells just don't produce enough mutant peptides that can be presented and recognized by T cells. And certainly there's evidence that within some tumors that there's selection against certain epitopes. There's evidence for that. And the other bit of evidence here is that more mutations in a tumor generally means a better immune response. The other hypothesis is that there's some other cloaking mechanism which is hiding the cancer cell. And so really the field is trying to test hypothesis 2 by combining lots of PD-1, PD-L1 inhibition with other immune modulators. And the immunotherapy trial I went on was just such a combination where we combined avelumab, the anti-PD-L1, with something called utomilumab. It's very hard for me to say, but at any rate, what this does is it interacts with a protein called 4-1BB on T cells, so 4-1BB, I guess, this interaction flips the switch and turns T cell division on. So you get more T cells, and the thought is that if you just have enough, they can overwhelm the defenses and kill the cancer. Well, that didn't work. So over the course of about 5 months, my tumors progressed beyond the sort of hopeful, maybe it's what we call pseudo-progression, where immune cells are swimming into the tumor and maybe they're going to defeat it, to just now this really is progression. We need to do something about it. So I returned to pemetrexed, and that explains the first gap. What about the second gap? So this involved the second surgery to remove a pemetrexed-resistant tumor, which was growing on my right lower lobe. We sort of watched it for a while, and it was getting bigger, and I was thinking, oh, great, radiation. And then Renato said, hey, we could take it out. And I was like, no, wait, we could take it out? We could do stuff with it. They say every cloud has a silver lining. So what can you do with a tumor? This is actually the tumor. And then key people in this process, obviously Doug Wood, the surgeon, Susie Densitz, the pathologist on hand, and Maribel Herr, who faithfully carted these specimens to these different folks. So, so Nick Crisp, he's my next door neighbor, right next to my lab. He's an immunologist, and so he opted to do, or he volunteered to do immune gene expression screening. Dr. Harold Sverdlow is a friend of mine from graduate school who is the director of the New York Genome Center. And when he heard, he said, hey, we could sign you up for a clinical trial to sequence your genome. Okay. And then I contacted McGarry Houghton and Pam Becker about the possibility of doing some high throughput drug screening against my tumor. I mean, we're going to have it out anyway. Could we get something out of it? And so what I'm going to do is just in the next couple of minutes tell you about the results of these cool experiments. I'm going to start with Harold. But first, I need to tell you a little bit about my family. So remember this issue of inherited mutations? This is my family tree, or at least a representation of it. My dad, you've met him already, and my mom, whom you haven't met. I alluded to earlier that she had cancer. She had ovarian cancer in her 50s, and then she had bilateral breast cancer 5 years apart in her 60s. Now, the other person of note here is my brother, who at age 47 was diagnosed with nodular lymphocyte predominant Hodgkin's-like lymphoma, which is a Hodgkin's-like lymphoma, if you couldn't have guessed. So when you see this sort of situation, if you know anything about cancer, you think, well, that sounds like an inherited breast cancer susceptibility syndrome. And that's what we thought. We'd been following down this road for some time, actually, even when I was in grad school, getting her tested at Myriad Genetics. And, but then I happened to run into these guys, because I was sequencing stuff with Tom Walsh, who's in the Department of Genome Sciences. And I told him this story, and he said, what, are you kidding me? Your mom's got breast and ovarian cancer. This is what we do. And so what they did is they enrolled my brother, myself, and my mom into their clinical trial. And they did exome sequencing on our germline. And the answer is that there is no BRCA1 or 2, but there was this mutation in MDC1. Mom is heterozygous for it. My brother carries a different allele of MDC1, and I have both. So I'm a compound heterozygous for a gene called MDC1. MDC1 stands for mediator of damage checkpoint 1. So the role of MDC1 is that it participates in double-strand DNA break repair. And this is a very complicated pathway that I'm not going to go into, but the key thing is MDC1 is down here at the center, and BRCA1 is right next to it. So BRCA1 participates in the same pathway as MDC1. Now, this does not mean that MDC1 is responsible for my mom's cancer, but it is a hypothesis. So this is a result of the whole genome sequencing. And what I want to do, first of all, is just point out that there is evidence for chromosomal instability in this tumor. And you're going to find this in lots of cancers for sure. So there's no evidence that MDC1 is there. But these are alterations in known cancer genes. So this is something which is frequently going on in that tumor. The second thing, I'm going to pivot, and I'm going to talk about some insight to pemetrexed sensitivity and the resistance of my tumor. So just point out that the structural variants in this case are in a cancer gene called STK11, serine threonine kinase 11. And that actually codes a protein called LKB1. Now, bear with me, LKB1 is part of a regulatory pathway. And what it does is it activates this complex here, called the AMP kinase complex, which then goes and activates this tuberous sclerosis complex, the TSC complex. And then what this does is it goes and it suppresses a complex called mTOR. mTOR is one of the central nutrient kinases in biology. And what it does is when it's rolling, it says, hey, let's make some ribosomes. Let's really make a lot of protein. This would be great. So I'm deleted for both copies, or disrupted for both copies of the gene which makes that, which means that the TSC2 complex is not going to be suppressing mTOR, and that can be driving my cell proliferation. This is probably at the trunk of my tree of cancer. So I was reading the literature, and I came across this interesting paper, which I don't have the reference for. This paper found that tumors that were driven through the same pathway, through the mTOR pathway, were exquisitely sensitive to the inhibition of the production of DNTPs, or NTPs. So this IMD-PDH is in the production of the guanine, the G in nucleotides. And so if you add this, what happens is that you get DNTP depletion, and replication stress, DNA damage, and cell death. So pemetrexed is an antifolate that interferes with the production of DTTP. So that's one of the nucleotides. And these are 3 abbreviations that might be useful. So the way that the DTTP gets produced is that in the early step, thymidylate synthase converts DUMP to something called DTMP. It does this using THF and DHF, which are tetrahydrofolate and dihydrofolate, respectively. Anybody take vitamins? Folate, right? Folate is an essential vitamin. And it turns out that this is important for catalyzing the production of DNPs. So in order for, and then pemetrexed is called an antifolate. So what it does is it interferes with thymidylate synthase. It basically takes the place of THF. So this suggests that pemetrexed in me is exquisitely sensitive because I'm driven by a TOR-dependent pathway. My cells are trying to make tons of ribosomes, and so they're sucking NTPs away from the DNTP pool. And now I add insult to injury by limiting the DTTP concentration. And so it's been known for a long time that limiting thymidine concentrations in cells is lethal, but this would just magnify the effect, sort of compound it. Now, the resistance. So it turns out in order for both pemetrexed and THF to work well with thymidylate synthase, it has to be polyglutamylated, which is where multiple glutamate residues, glutamates and amino acid, get added to the tail of the folate. And lo and behold, within the whole genome sequencing was a deletion, an in-frame deletion in the FPGS. And if you go and look, it's in the active site of the enzyme. And if you go and look in the literature, this has been described. So that's how my tumor was escaping. Pretty cool. So I think one conclusion from this is that my tumors likely remain exquisitely sensitive to DNTP concentration, and that could potentially be a future therapy. So I'm going to just talk about this exciting project. So this is really a collaboration between these two, not just with me. So a PDX mouse, that means patient-derived xenograft. And what happens is that you embed a bit of tumor into this sad little mouse. It develops a bump. And then you isolate that tumor, disaggregate the cells, a miracle happens, you plate them, and then you give it to the Quellos high-throughput screening facility, which is down at the South Lake Union. Pam screened 170 different drugs, which were part of a drug panel that she had for multiple myeloma. What I haven't told you, and it's really important, is that Pam has been doing this sort of empirical approach to treating leukemia and myeloma because she thinks, and I agree, that an empirical approach to looking for chemotherapy sensitivity has got to be better in some cases than just trying drugs at random, or drugs that you think will work. And that kind of goes back to this idea that each cancer is different and on a different evolutionary trajectory, maybe related but different. So they did this screening with viability, and this is the top 25 candidates. And I've highlighted them according to different classes. So what you can see is there are some genes which are, or some compounds which are HDAC inhibitors. That means they affect a protein called histone deacetylase. And then there's protease inhibitors, there's microtubule inhibitors. These are things that are important for cell division. There's a whole list of things. I don't have time to bore you with them now, but the main point here is that there's a lot of chemotherapy options to pursue when the time comes. And these are not necessarily, some of them are, but they're not necessarily the ones we would have gone with. We wouldn't have gone with HDAC inhibitors if I needed to shift to a different chemo. So the last of this little bit, and I realize I'm pushing on towards time, but is the expression of immune-related genes. So my next door neighbor and this absolute ray of sunshine, Dr. Zhao, who's his postdoc, what they did is they isolated mRNA from my tumor. They then assessed what the levels of transcripts were in select immune-relevant genes. And this is their experiment. So it is not a perfect experiment. They didn't have normal lung tissue, for instance, to use as a control. They used liver, and they used virus-infected liver. They just threw it alongside of their experiments, basically. But nevertheless, the results are interesting. So PD-L1, which is the ligand for PD-1, its expression was thought to be low. 4-1BB, also low. Of course, that is more a reflection of T cells that might be in the environment. So the absence of that is maybe not too surprising. This kind of explains maybe why I didn't respond to the first immunotherapy. And then over here, CD47 is elevated relative to what you'd find in liver. So I'm going to focus in on this because this turns out to be kind of a nice part of the story. It just so happens that last gap over here corresponds to an anti-PD-1, anti-CD47 clinical trial that I've been on. So CD47 is a don't eat me signal. It interacts with macrophages through the SIR1A protein and says, just chill out. So the hypothesis is that part of the reason why there's this envelope of protection is that CD47 is preventing priming of the immune system. And that if you just interfere with that and at the same time interfere with PD-1 and maybe through some crosstalk between these two cell types, you'd really get some traction on the cancer. So 2 months in, October 2018, I was ready to quit. My tumors were getting large. This isn't even the largest. I had one that went from 1 centimeter to 3 centimeters by 1.5. It just didn't seem good. December, things started to change. That 1 centimeter, that 3 by 1.5 went down to 2.5 by 1. It started getting noticeably thinner, and there was evidence for immune infiltrate present. And now here we are, April 2019. I just had a scan in July. It essentially is the same. This response seems to be holding. Now, there are small tumors that have completely gone. Tremendous. We just don't know really what this is. Is it just leftover immune cells? Does it represent residual disease? We won't know for some time, but it's not like I'm going to go off. I'm going to stick with it. So that is basically the end, but I'd like to leave you with some thoughts. The first of all is, I'm going to reiterate what I said at the beginning. This is the best time in history to have cancer. Don't lose hope. Chances are you know somebody, or will, or maybe you're going to need to know. For an evolving foe, we need to awaken an adaptable champion, the immune system. Chemotherapy still has a role to play, especially an approach that recognizes the immense diversity of cancer and seeks to encounter it with empirical measures of drug sensitivity for individual tumors. Man, did I write that sentence and not actually read it? I think that's probably true. The last sentence, though, be kind and generous with your time. All those smiling faces that I showed you, picture after picture of incredibly generous, amazing people. You'll make an N of 1 something more. Thank you.